Yes.
Great. Okay. So welcome to ImmPort. This is an introductory webinar, as Dr. Sanchita presented, introduce us, and we're going to go over ImmPort and give a high-level overview, and hopefully you'll learn a lot and come back for more webinars as they're presented later on in the year. So this is the ImmPort ecosystem. ImmPort is composed of 4 applications. We have an application to capture and curate data. It remains in a private embargoed workspace until it's ready to be shared, at which point it's moved over to a shared application so that it's easy to find, access, download, that sort of thing. There's a data analysis platform based upon the Galaxy framework that allows folks to have a graphic interface to command line open source flow cytometry, mass cytometry tools. And we have a resources application with many tutorials, lots of documentation, listing of publications, that sort of thing. All of this is possible because of the excellent work of a fairly small group of people. And so much obliged and many thanks to the ImmPort team for doing a really cool job of getting a lot done in a reasonably small amount of time. So ImmPort is developed and funded by the National Institutes of Allergy and Infectious Disease, specifically by the Division of Allergy, Immunology, and Transplantation. And ImmPort's purpose is to collect and share immunological research data from both mechanistic studies in animal models, also clinical studies, and also clinical trials data. So our goal is to capture data, annotate it well, and then share it according to the FAIR principles. ImmPort distributes data funded from multiple sources. Our primary source is NIAID, but we also get data from other NIH funded resources, including the NIH Foundation, NCI, March Dimes, Bill and Melinda Gates, and several other non-US based funding agencies. So this overview will highlight how to find and access ImmPort data. And as I mentioned, there will be follow-up webinars to go into more detail for particular topics. We're going to go over the data model real quick and understand how it's organized. We're going to discuss finding and downloading of data and a little bit about the future activities that we're working on. So this is a graphic representation of the ImmPort data model. It's actually an interactive model on the webpage. It has a couple of features that I want to highlight. The model is intended to provide a consistent format for descriptive and results data to make it easier for the users of the shared data to understand how things are organized and where to find them. And it's also intended to provide a standardized definition of the terms, a standardized representation of terms, so that folks are understanding and have a consistent nomenclature for the terms that are used. There are lots and lots of synonyms in biomedical research, and we try to represent the canonical term whenever possible. We use a variety of ontology resources and also the NCI thesaurus, clinicaltrials.gov. These are our reference sources. So the data is organized by study, and within a study, there is a description of subjects organized by arms and cohorts within a study. There's also a description of the planned visits, so the temporal sequence of events, the timeline of the study details. Subjects may be described and measured in terms of both sample or non-sample based measurements. So if it's a non-sample based measurement, it might be a questionnaire, so a case report form that captures BMI, autoimmune severity index, that sort of thing. If it's a sample-based, then samples have a temporal description. When was it the sample drawn from the subject or in the course of the study? The type of subject, the where from the subject the sample was drawn. And then the samples can be assayed either via a lab test, a clinical lab test, which is under federal, regulatory controls, so LabQuest or that sort of LabCorp sort of lab test. And then also they could be assayed by experiment measures. So we capture lots of details about treatments or reagents. And we understand that the format or readout of the assays are different for different assay types. And so we have customized formats for capturing the many types of immunological assays from ELISA, flow cytometry, gene expression, proteomics, that sort of thing. The model on the web page is interactive. So if you click on a node or a concept, it explodes out to provide details of the attributes for that concept. So that may include descriptive elements for the term, also links to other parts of the data model. So reference sources, how a concept is linked. So for a subject back to an arm or a subject to a sample. The import shared data site allows you to search via a variety of means, and this is primarily a graphic user interface. And I wanted to highlight a couple of those search mechanisms that makes it hopefully easier to find data, to get an overview of the data that's available, pique your interest, and perhaps encourage you to download and hopefully reuse the data. So one of the summary overviews is a data summary matrix, and allows you to do a pairwise comparison to know what sort of data is available for download. So for instance, we have a listing of the study days that are available within all the different studies in ImmPort, and then the assay type. So you can understand, for instance, if you're doing HAI analysis, whether how many studies have day 0, day 3, day 7, day 28 measurements after vaccination. There's also a pairwise combination. So if you're interested in finding studies that have both flow cytometry and ELISA, or gene expression and CyTOF, you can find it easily here. So if you clicked on one of these nodes, it would take you to a list of studies that satisfies those requirements. This is the bubble graph. Again, it's a pairwise comparison or listing of the data. It's an overview and allows you to see what sort of assay types might be available within a given research focus. And there's a variety of different pairwise comparisons that are available from the drop-down menu. This is the ImmPort study search page, and it has both a facet search capability and a text search capability. So for the facet search, if you click on the zoom button, then you can expand the pie charts, and you can choose one of the or more of the slices to filter on those studies that satisfy your interest. In addition to the pie chart, there's also a drop-down listing for each of these facets, and it's a text-based. You can select the terms, one or more, and it will filter the studies that are present in the study catalog below. You can also apply a free text search. So if you're interested in influenza, yellow fever, transplantation, anything like that, you can use those terms within the free text search field. So once you've applied your filters, then the study catalog table responds to those filters and displays the relevant results. So for instance, I typed in COVID, which is also a faceted search term, but this is a free text option. And we have several studies that have COVID as part of the descriptive elements, either in the title and the description, something like that. Right? So if you click on this link here, it would take you to the study detail page. There's more information about the subject demographic summaries, the listing experiments or lab tests or other results that are available. If you click here, you can get to the download page and actually get access to the data. If you click on this little arrow, it provides more details about each of the studies within the table, so you don't have to go back and forth if you're not interested in doing that. Currently, or by default, we have 6 columns selected for display in the table, and you can do greater than 20 studies, 20 columns in the table if you wish. You can export the table as a pre-filtered listing of studies of interest to yourself to save later and to come back to. You can also choose to download all studies, and it'll take you to the download page for all the data sets that represent all the parse data for the shared data sets. There's an API endpoint option. So for those of you who are more interested in command line interfaces as opposed to a GUI interface, we have that capability as well. So when you click a study to download, then you'll get actually you'll be focused on a study detail, much like this is. And if you can go back and forth within the data browser to see a listing of the study accessions that are available for download. Within each of the study accession listings, there is a listing of the files that can be downloaded. And this provides a quick overview of what's available within a study. So there may be multiple types of result files, flow cytometry, mass spec proteomics files, that sort of thing, custom file formats of some nature. Protocols are available, obviously. We provide two options by default for download. One of them is a MySQL database dump. We also provide the schema and the loading files for creating the schema and loading it into the schema, into the MySQL instance. And we have a tab separated text file format for folks who want to use it in tools such as Python or R. As I noted, there is an API option for folks who are command line interface preferred, and we have a large numbers of endpoints. A lot of them are based upon the assay type, but also at the study level, the subject level, that sort of thing. We have a number of tutorials available for you, as well as other resources, documentation and all that. And I want to highlight that we're hoping in the early spring or summer, we will be able to release a version of the tutorials that are interactive. And so they are going to be hosted in a Jupyter Hub platform, and you'll be able to run the tutorials. You could make edits and rerun them and actually see things run in real time, so that you have a feel for what are the possibilities that are available to you if you use R or Python scripts to access, to process the data and query it. So the tutorials, the current ones, as well as the future ones in the Jupyter Hub platform, allow you to explore the data model, find out what's there, to pull data together, to aggregate data sets, filter them, and do analysis. So this is an example of a tutorial that Dr. Hugh created for the Focus 2020 workshop. And I wanted to highlight that the UCSF team is again doing a workshop at Focus 2021. It's called Big Data in Immunology. We encourage you strongly to register and participate in it. And they'll be going through tutorials that explore import data and highlight how it could be useful to you. And hopefully you will get publications and you will get grants and you will say good things about import, and that it was actually a valuable resource for you to explore. So with that, I'm going to allow Dr. Schaefer to do the presentation, and I'll run the slides. Thank you very much.
Thank you, Patrick. I hope people can hear me. I'm Henry Schaefer, and I'm a data curator on the ImmPort project. Here I'm showing on the screen, you can see the ImmPort landing page that has a link to the ImmPort private data page that you access when you want to upload data to ImmPort, as well as links to the other resources that Patrick mentioned. Next. So this is the ImmPort private data page. This has a link to the help desk. We pride ourselves on answering questions. We can answer questions about not only submitting data, but also shared data as well. You can see I've put on here on the slide a list of the data types that we have data loading templates for. If you can see if your favorite data type is here, either your favorite data type for uploading or for downloading. You can see at the top we have molecular mechanistic assays, many data types for that. And then on the bottom, you can see more clinical data types, such as assessments and lab tests. Okay, so we have both mandated and non-mandated uploaders, as Patrick mentioned. Non-mandated uploaders include Bill and Melinda Gates Foundation and March of Dimes. And our mandated data submitters are mandated through NIAID to submit data to ImmPort. Our data upload templates do a lot of the enforcement of the data requirements in ImmPort. We forbid names, dates, identifying locations, that sort of thing in ImmPort metadata. So those are forbidden from upload templates. Uh, any dates that are in study metadata have to be converted to study days, and all original identifiers that are used to link together data and upload data, those are all converted into import accession numbers for subjects, for biosamples, for experiment samples, and none of the original identifiers are ever shared. Okay. Next. So we where this usually starts for our non-mandated data uploaders, usually they contact us through the help desk, either before or after creating an import user account. We will ask you, so as we mentioned, import hosts immunology data, and this right spans a large range. Everything from multidimensional molecular data sets to clinical trials that have primarily, lab test and assessment data, questionnaires, that sort of thing. We also have cancer immunology. We have preterm birth, metabolomics studies. We have a wide range, but we focus on immunology. And so if you contacting, we encourage you to contact us to ask if you want to share immunology data. We encourage you to contact us, and we'll ask you some questions so that you understand. We want to ask you about what your expectations are in terms of schedule. If your upload is to satisfy a publication requirement or to satisfy a grant requirement, and your expectations about the schedule. We have data releases every quarter at least. We'll ask you some questions also about funding so that we can properly annotate your study before creating a workspace for you. So a workspace is where you begin uploading your data. And you can see here, I've taken just a segment of the data model that Patrick showed. And this is the first steps. Just wanted to show the first steps for creating a study. And then I also included the next step is usually creating subject records. Okay. So what happens is when you upload the basic study design and the protocol, an import study accession is created. So if you need a study accession for a publication, for example, this is the parts here in orange and red. That's the initial steps that are required to create the study. Let me show you in the next slide, Patrick. So here I wanted to just show you some of the data elements, what's required to get started. You can see here in the basic study design I've listed the data elements that are required in black and that are optional in red. And if you go to here, you see on the left side of the screen, you see an example of an import shared study, and you can see that those same data elements are mirrored in our shared study summary. If you want, and then I've also listed here the data elements that are required for the subject human template, both required and optional. We also have a subject animal template for animal data. To see those data, to download those, you do need to log in because you will see the row specific data. But that will, if you want to see more about how that data gets organized and what that looks like, we invite you to download data and take a look. Okay. Patrick, can you give me the next slide? Thank you. So one of the principles that ImmPort works on is we try to harmonize our data to make it more useful for reuse. For our data downloaders, we try to harmonize it and organize it so that looking across studies, we have a common data model, and um, studies are more easily compared. Okay, so what you're seeing here is this is an Excel version of the human record loading template, and you can see well, the data elements that are required and optional, and you can see in immune exposure we have a draw. This is the Excel template. These are also available in text for those who are more, want to do this in an automated fashion. But the Excel templates have some useful features, such as the drop-down lists here. So what you have here for the subject's human template, for the immune exposure, for the material reported, what we do is we have a preferred vocabulary, and this is populated with exposure materials from both the vaccine ontology as well as the NCBI taxonomy browser. You can see it's important to be, to clarify right here between infections and vaccines. So these immune exposure fields are optional. They can also be updated if you're loading data. Very often in ImmPort, you can, if you need to go back and re-annotate things to correct records, either before or after sharing, we can do that. Okay, next. So here's another example of how ImmPort uses ontologies to harmonize our data. Here you can see disease reported. We bring in, you can see here, there are diseases from disease ontology, and then there's also records from NCI thesaurus that are not diseases. They're such as the one at the bottom there, heart transplantation. We capture it in the disease reported column, even though that's not, it's more of a medical history or condition, but we capture it in the disease reported column. Okay. Next. So one of the things ImmPort tries to do is in order to harmonize our data. Here we see we have our preferred vocabulary. This is a reagent template for CyTOF, which is a kind of flow cytometry, and we have a list of hundreds of analytes in the preferred vocabulary list. We have a lookup table that we update when needed to add new terms here, and then on the bottom, what you see, this is a shot from our shared data, and it gives you an idea of how the ImmPort database is organized. You can see in the red box what we have. We have analyte reported, and then on the top row, you can see there that what we do is we populate the analyte preferred with the preferred vocabulary, and that allows us to capture the original reported data, but then also to harmonize it. And this happens both before sharing, but then, when these features were implemented, right, our old data was harmonized using this same mechanism. All right, next. ImmPort. So this is our gene expression array upload template, and it shows you another feature of ImmPort. We try to leverage federated data. So we will link to, whenever possible, we will link to data that's captured elsewhere. Here you can see in the dropdown, we will link to geo annotations as well as dbGaP annotations. So if gene expression data has been submitted to geo, we just require that that geo accession, and then we create an experiment sample record around it. And you can see the experiment sample is there. Those required and optional columns are in blue. And then the biosample record, which gets created with the same template as needed, if needed, is there in red. And you can see what we do is we capture, we link that biosample to a planned visit and also to a study time collected. And we can answer questions for you. If you have ideas, the ImmPort data model is very flexible. So it can be used different ways in terms of how do you want to define your biosample. Someone might want to say that they want a bunch of experiment samples linked to the same biosample, and they'll call this biosample whole blood, and that's how they'll want their data organized. And someone else might say, I want to have my whole blood biosample separate from my serum biosample. Maybe they were collected at different times within the day, and you want to set up separate biosamples. Maybe, but maybe not. Maybe you want to just have one biosample so that when you're looking at these experiment samples, they're all linked together because that's how you want to represent your data. So that's why we encourage you to contact us. We have extensive documentation, but we find for many data uploaders, the fastest way to get started is to talk to us about your specific data and to get you started. And next, Patrick. ImmPort is a recommended data repository recommended by Nature Scientific Data, as well as Public Library of Science. As I mentioned, we release data quarterly at least, and this is when we have a data release. This is when we apply a lot of quality control scripts. This is when we do some of our harmonization. It's kind of like a publishing process. We don't just let studies go out in the wild. We make sure they don't contain any personally identifying information, and make sure that they don't. A lot of these things are database checks to make sure that things are properly organized and not misaligned. The records are correct. So until we release you data, the data is held in the private workspace, and it's restricted to specific user accounts so that you can upload your data and add users who want to take a look at it before it gets made public. When it is made public, we assign a DOI to the study so that it can have a link that will not change. It can be updated so that your DOI will always point to your data set within ImmPort. And with that, that's all I have, and I'll be happy to answer questions at the end of the hour. Thank you.
Right, thanks, Henry. Hello, everyone. My name is Zichen Hu. I'm a researcher at the University of California, San Francisco, and a member of the ImmPort's team. And next, I will give an overview of the tutorials that's available for using ImmPort's data. So first, I want to mention that the ImmPort website provides a wide range of tutorials, and these tutorials are available at the resource page, followed by the tutorial tab. And you can see here the imports. In ImmPort, we provide tutorials on how to use ImmPort API, how to download and upload import data, and also I guess the most useful part is tutorials for data analysis. Next slide, please. So in addition to the existing tutorials on the ImmPort website, we also provide most up-to-date tutorials on GitHub pages, such as this one you are seeing. If you want to find it, just search ImmPort Focus 2020 on GitHub. And this tutorial is created last year for our Big Data in Immunology tutorial in the Focus conference. And the GitHub page is a very self-contained tutorial, which includes the already downloaded data as well as a Jupyter notebook. So if you download the whole repository and start running the Jupyter notebook, you will be able to run the tutorial without needs to download data from ImmPort's. Next slide, please. So in this presentation, I'm just going to give you a very brief overview of what this tutorial will offer, and I encourage the audience to check out the GitHub page and run the codes using your own computer. So in this tutorial, we show how to analyze a clinical trial data. So this clinical trial data is from study SDY-824. The title is Anti-TNF Agents in Rheumatoid Arthritis. So if you look at the later panel, we show a snip of code that kind of display the arm design or the cohort design of this study. So you can see that in one cohort, they are using one type of anti-TNF drug at NASCEPT, and another cohort, they are using another anti-TNF drug, adalimumab. So this study is to compare the effects of these two anti-TNF drugs. Next slide, please. So first, the tutorial will guide you to navigate through the ImmPort data structure. So this data structure overview chart may seem intimidating at first. However, for most study, there's a kind of main trajectory to follow. That is, first we have a study, then we have multiple arms or cohorts in the study, and each arm or cohort contain subjects, and the subjects can donate or contribute biosamples, and these biosamples can be linked to a lab test and other experimental data. So for most study, if you follow this path, you can kind of manipulate and collect information you need in ImmPort database easily. Next slide, please. So in this tutorial, we provide examples on how to navigate this data structure. So we have different code snips for each step. So first, we will identify subjects in each study arm. And as you can recall, this study contained two arms, treating people with different anti-TNF reagents. Next, we need to find some information related to these subjects, such as gender, age, and race. In the next step, we map biosample to the subject to identify which blood samples come from which subjects. And finally, we combine the biosample with lab test results. So using these code as an example, you can collect arm information, subject information, biosample information, and lab test results from the ImmPort's data structure. Next slide, please. Then the tutorial will provide examples on statistical analysis using the data you just collected. So in this particular case, we are running a mixed effect model. So the lab test value will regress on the time of the study, the type of drugs they receive, and also controlling for variance based on subjects, which is treated as a random effect here. So you can see this particular code snip provides a summarized table. Here, the p-value is how significant the lab tests differ between the two treatment cohorts. And you can see that the neutrophil number seems to be significant different between these two drugs. Next slide, please. The tutorial also have lots of examples for data visualization. So on the top row, you can see we are comparing the age and the gender distribution of these two cohorts. And we can see that this study is very well designed, and the demographic information of these two cohorts are comparable. And on the bottom row, you can see we visualize the trajectory of lab test values in these two arms. And for most of the lab tests, they are kind of similar, or there's no difference between these two cohorts, which is as expected, since both of them have the same mechanism of action. However, as consistent with our statistical analysis, the neutrophil number is very different between these two drugs. So this could be an interesting finding, reanalyzing this clinical trial data. Next slide, please. So yeah, as I mentioned, this is a tutorial we provided in last year's focus workshop. And in this year's workshop will be in June 8th, and we will provide a updated or new topic of workshop. And I encourage people who are interested to attend this workshop this year. Yeah, this will be the end of our presentation, and we'll open up the Q&A session to discussion and answer any questions.
Thanks, Ji-Cheng, Henry, and Patrick. Great presentations. So as Ji-Cheng said, let's move on to the Q&A. I see on the Q&A there are two questions. I'll just read it loud. The first question is from Janet Dell, who on a study team is typically the point of contact for study submission to ImmPort, like the PI or the statistician or the team?
So I can take that first question. Hi, Janet. Usually there's a data manager designated for a research team. It's not usually the PI. And sometimes it may be the statistician, but it's somebody who has access to a lot of the data, potentially not all of the data. So there could be data sets that a team has and maybe part of a publication that are not sent to ImmPort. That's up to the research team and their program officers' expectations.
And the second question is, and what is the general time point when this occurs? So do we have a timeline for submitting the data to sharing the data?
Oh, a most excellent question that comes up a great deal. Some folks start sending in their data when they're starting to write the manuscript, because then they have a good idea of what set of their data, subset of their vast amount of data will be published. In other cases, folks set up a workspace and put in a study outline at the beginning of a project, and it might be 6 months, a year, 2 years before they actually start submitting data to ImmPort. It really depends upon the research team's goals and their program officers' expectations.
Any other questions? If you prefer to speak, please raise your hand and we can turn on the mic for you.
Hi, this is Quan. Can you hear me?
Yes.
Oh, okay. First, congratulations for the first science meeting. I have a question for the panel, but probably mostly for Patrick. I think the data model has been presented under certain context, but I do have a general one considering the heterogeneity of the data generated from various resources, but also various reuse scenario by the downstream potential users. And also a third one, which is the internal need for a data model by the curators of a repository. Do you think it's still possible to come up with a overarching, but rather also simple data model with essential metadata structure that would somehow satisfy, the three masters, the original resources, the repository curation, harmonization, but also mostly downstream reuse cases?
Excellent questions. We have this discussion with our data providers, our contract office representatives, our standards working groups, and our science experts on a regular basis. So the data model that was presented is what you might consider the gold standard. So it supports a lot of description of things and very well described. There's also other levels, so silver, bronze, tin, if you will. And so we customize the annotation expectations and the annotation capture and the type of data, the format of the data, based upon the researcher's needs and expectations. So to your question, Quan, absolutely. We have done everything from having just a study outline that has high-level descriptive information, and the results are provided as study files, and they have very light curation, if anything. So there's the custom format provided by the data providers, all the way to we actually parse the results or many of the results into the data model. So wide spectrum of capability customized to the data provider's needs.
So if I understand, it seems to me that by allowing quite a lot of flexibility in terms of populating the entities in the existing data model, we can, at least in most cases, readjust our curation and harmonization effort, therefore more or less customize the data model use, right? In various situations, I guess, yeah.
That's correct, yes, absolutely.
Okay. Yeah, so let me just quickly a follow-up question for Ziqian. So Ziqian, based on your actual downstream reuse of the data structured according to import data model, and then a lot of studies and entities associated with the studies. So in general, I think this is more like sharing experience. So in general, did you found the way the study has been shared, structured, it's under-organized or overly organized?
So my my experience is there's a learning curve to kind of learn how to navigate the import data structure, because there are, for example, in my tutorial, it requires 4 joining to collect all the data I needed for the statistical analysis. This one aspect is this kind of flow is standardized for all import studies. So once you overcome the learning curve, the procedure is very simple. And also we provided multiple tutorials on how to do this. But there is an aspect that new users will maybe feel a little bit lost trying to find where things are in the import database. So I'll provide one example is the import data structure kind of puts puts gender and race information at the biosample at the subject table, and the age information is with the biosample, and this makes sense since a subject can participate in multiple years and his age will change. But for a new user just trying to find the demographic data, it's kind of a little bit strange to trying to find, race and gender information in one table, but age information in a totally different table. And people may spend time trying to look for this information.
Okay, thank you very much.
Okay, so then it's a great segue to, again, for Jicheng, there's a question about, are these tutorials available for secondary data analysis?
So I first want to clarify what does Amber mean by secondary data analysis. So my understanding is secondary data analysis means you analyze the data and they're trying to find things that's not reported in the original paper, or you're trying to answer a question that's not the main question answered by the original study. I think we have several tutorials trying to kind of repurposing the data. The tutorial I just went through is an example of secondary data analysis, since the main goal was to look for B cell differences. And here we are kind of going over all the lab tests and see what else are different between the 2 drugs. We also have several tutorials available on imports. One example is we use a tool called Metacyto to perform meta-analysis of flow cytometry data and we compile data from more than 10 studies and do the data harmonization and try to see what immune subsets are different between different ancestral groups in the US population. Hope that answers your question.
Any other questions? If not, then thanks everyone for attending and for your participation. So stay tuned, hoping to see you all at the next webinar, which is scheduled in June. And we will be sending out announcements. And we plan to, for the future webinars, we will take a deep dive into the specific areas of data exploration, data reuse, and repurposing, and the tutorials for data, how to explore the data. So we'll take a deep dive in the future webinars. So again, please stay tuned. And thanks again for your participation.
Thanks, everyone. Bye.
Bye.